some which seem to exacerbate disease. Quite how that's happening. These RNAs are presumably also picked up through the capsids, so they just get encapsulated. They're replicated by the viral RNA-dependent RNA polymerase. So you get lots of these. Turns out that these three residues down here at this end of this particular RNA, which leads to a nasty phenotype, much worse disease. If you change these nucleotides, then basically all disease goes away. Again, very open question about how these things are actually functioning here. So lots of questions that people are still trying to answer. So I want to talk a little bit about CCMV, cowpea chlorotic mosaic virus, because this virus is, like a lot of these virions, very, very stable. And it kind of has to be stable if it's floating around on aphids, and it's got to be hanging out inside the cytoplasm of your plant cell in order to be able to get transferred later. But if you got a really tough virion, how does it let its genome out, or its segment of the genome? So what happens in this case is, again, it's just like the kid's toy. It's compacted on some cases, but as soon as you change the pH or the ionic strength, which is going to be different outside of your aphid or inside the plant cell, it opens up. And this is a completely reversible process. And turns out you can just express the capsid protein from CCMV, and it will make this particle, which will undergo this reversible process. So really amazing nano-sized process. These are about 25 nanometers in diameter in terms of going back and forth here. Normally what would happen is the virus genome is going to be, or should say the genome segment, comes out here. In the case of CCMV, it's actually 2 segments rather than 3. And then will be translated. You'll make more virus particles. But a couple of very smart people, way smarter than me, including one of my postdoctoral advisors, said, well, if RNA can be coming in and out of here, maybe you can put other things in and out of there. And the first one they thought of was metals. Now, why metals? Metals are, of course, very interesting for all kinds of different things, but in this particular case, what they were looking at were ferromagnetic metals. And one of their main sponsors for this project was Toshiba, who were really interested in how do you raise the capacity of hard drives, because what is on a hard drive is a bunch of magnetic particles. And it really depends on how small you can make those magnetic particles. Well, it turns out you can take CCMV and put magnetic particles inside them. You open them up, put the magnetic particles inside, put them back together, and then because you can get really ridiculously large numbers of virus particles, virions, then lay these down on basically, again, what would be a hard drive, burn off all of the capsid proteins, and you've got these perfectly spherical 25 nanometer magnetic particles. So basically raises the density of magnetic storage by about 2 orders of magnitude, just by using these virus capsid shells. They're also very interested in using these for magnetic resonance imaging. If you can engineer the structure of the protein on the outside so that they interact with, say, a cancer cell, you put a contrast agent on the inside. This could be really, really useful for MRI. In fact, they've patented some of this, and some of this I think is even already in the clinic. Some chemotherapy drugs, same idea here. You can put RNA in and out. You can put whatever else you want in and out. You've got this reversible condition. Chemotherapy drugs, lots of ideas about how this is happening. Again, Mark Young is the guy I was working with at Montana State University, although I wasn't working on this project at all. And Trevor Douglas, who was at Montana State, has now moved to Indiana University. Interested in some more applications here. Particularly Trevor's doing some really cool stuff as far as this is concerned. Mentioned hot viruses other than the ones we're working with in my lab. But this is one of the plants that you see when we are literally going out into the field, into the hot springs, and looking for various different viruses. It's a plant called Dicanthelium lanugnosum, also known as Texas panic grass. I much prefer that. Really wide-leafed grass that you find at ridiculously high temperatures. This is not Fahrenheit, this is Celsius. So these plants can grow perfectly happily up to a little over 50 degrees Celsius, which is pretty darn amazing. And in fact, we can go out in the field when we're in some of these hot spring areas, Yellowstone, Lassen Volcanic National Park, and just by seeing where the panic grass is, tells you what the temperature of the ground is. It's really pretty amazing. We can stick our thermometers in there, you get 50 degrees. One thing that was also being done at Montana State when I was there, it turns out that this grass can only survive at those really high temperatures when it's infected with an endophytic fungus. And so this is the endophytic fungus right here, Curvularia protuberata, found in lots of grasses, but this particular one is found in that Dicanthelium. But it's not just only when they're infected with fungus. What kind of class? What's the name of this class? It's a virology class. So it turns out that it only gets thermal tolerance if the fungus is also infected with a virus. And so these are the data down at the bottom here. So normal, infected with fungus and virus. Here, this is without the fungus. This is, and here is without the virus. So these guys are perfectly happy at 50 degrees. This one has no fungus, but this one has fungus and no virus. So you have to have fungal infection and virus infection in order to get this thermal tolerance. This is a cartoon, basically, just of how this works. So here's our Dicanthelium when it's got happily infected with fungus and with this Curvularia thermal tolerance virus, CHTV here. On the other hand, they're not colonized, they're really unhappy. There's a nice review article that Marilyn Rusnick wrote, in fact, the same author as the chapter here on this cucumber mosaic virus. So this is one of my favorite viruses that I don't work with because it's giving thermal tolerance. How it does this, there are a number of theories, but nobody really knows what's going on there. So it's not really a plant virus, but close enough. Real plant virus, on the other hand, tobacco mosaic virus. So this is the original Contagium vivum fluvium. This was found by Ivanovsky and Bayerjink, probably independently, but from each other. And the data are really pretty good on that. No one was reading the Russian in one case, and nobody was reading the German in the other case. I just pulled up this particular example here. Tobacco mosaic virus. Anybody know what this plant is? Is it tobacco? It's tomato, exactly. So tobacco mosaic virus actually infects tomato also really well. So last night, digging through the internet, pulled up Bayerjink's paper from 1898, Martinus Wilhelm Bayerjink, and I just love this quote. I'll let you guys read it. I wish I could use these things. Yeah. Incontestable experiments. I would just love to have an incontestable experiment that I could publish, or even to write any of this thing up here. This is the link, in fact, to the whole thing if you want to take a look at it. But I read this paper. It was really amazing, the stuff that he was already showing in 1898. He uses virus as well, so it's not just Contagium vividum fluvium. Shows, at least has some data, that it's only able to replicate in growing plant tissue. So he immediately, or say not immediately, but makes that connection that the virus can only replicate in something else that is already growing. And he shows heat stability. So he did experiments going up to 40, 50 degrees Celsius and drying out the virus. It was still infectious. So again, 1898, he's already making a number of predictions about how this is working. They had the diffusion experiment, which is probably incorrect, but just really amazing that that was what was happening there. So first virus as well, that in and of itself is probably not enough to write a book about. But people got really excited about tobacco mosaic virus, mostly because it was this really simple structure and something that people could work with. And again, contagium, vivum, fluvium, vivum, alive, but a particle. So we've got these alive particles. Is it really alive? Is it a particle? What's going on here? So one of the major people who's working on this, Wendell Stanley, got the Nobel Prize in 1948 basically for crystallizing TMV. They got the first crystals in 1935. Wendell Stanley was at the Rockefeller Institute, got his Nobel Prize, and Berkeley said, hey, we need a Nobel laureate. We'll hire him. And then we'll name this building after him. And so this is, or say this was Stanley Hall at Berkeley. And this window right up here in the corner, right behind that is where I did one of my rotations for my PhD, in this building. Giving you a bit of an idea, this building doesn't look like this anymore. Now it looks like this. It was completely rebuilt in 2007. Again, nothing else is left here. This is the new Stanley Hall down at Berkeley. It also tells you that they have a little bit more money than we do for putting together some of these things. Oops, one too far. Eek! Uh-oh, I was encountered a problem. Okay, fine. No, I don't want to send anything here. Let's see if we can restart it here. That's what happens when I edit. You know, we're talking about me editing my lectures from year to year. Obviously, this was a problem. So I should never change a running machine or whatever we should say here. So where were we? So this is the structure that was originally, not originally found, much later found after Stanley was able to crystallize the virus. Let's look at it here. What's going to work? Yes. Okay, good. So this we've actually looked at before. This is the really classic helical structure, tobacco mosaic virus. All of these proteins bound on the outside of the RNA. This is a naked virion, so the nucleocapsid doesn't have an envelope around the outside. The protein itself is here. It's basically a 4-helix bundle with this loop at the end here. I'll give you one guess what kinds of amino acids you have in this loop. These are going to be the positively charged amino acids, which then hook up into the RNA here. I'm going to try and go to this link, and everything may crash abysmally, so we'll see what happens. This is a link to the protein database and the tobacco mosaic virus. Again, this is where I got that structure from. This is a structure now looking at just 3 of those individual rings, and we're going to see if we can get this to work in JMOL. With any luck, it will come up. You can now use this and turn the structure around, zoom in, zoom out, rotate. What I'm doing is take a look at the side here. So there you can see really nicely how each of these rings is slightly too big to go all the way around, and that's what's giving you the structure as it's wrapping around this RNA and packaging the RNA all the way up through the top here. I can spend hours playing around with these things. But that's that loop here at the end, which is sort of looping around the RNA, giving a little groove in which the RNA is being packaged. And if we zoom in a little bit here, no, that's the other way to go. No, zoom out. Zooming all the way in. Okay, now we can't see it anymore. It's all gone. Okay. My computer's just too old here. We can spin it around. But what I wanted to show was those helices, the 4 alpha helices, which are sticking together, and then each of those are all stacking on top of each other in terms of the rest of the helix. We could talk about pandemic flu for 10 minutes, or we could go outside in the sun and pretend it's the weekend. Good. Okay. See you on Monday.